A very good evening to everyone. Let's crack CSIR-UGC NET together. Unacademy is one of the India's largest platform for the CSIR-UGC NET examination. So before beginning, let us have a small introduction about myself. I am Farida Bandookwala and I am pursuing my PhD from DAVV, School of Biotechnology, and I have done my BSc, MSc and MPhil in Biotechnology. I've qualified MD/SLET examination in 2018. I have a teaching experience of 10 years. Then what you will get with the Unacademy is if you're going for this plus subscription or you're just looking at Unacademy for the preparation, then what you'll be getting daily live classes, you'll be getting structured courses, you'll be getting live tests and quizzes, and you will get unlimited access to these quizzes. Now, what is so special about these things is in the live classes you can ask your doubts directly with your educator, you can chat with them and you can engage in the discussion whichever is going on, and you can ask your doubts and queries at that particular time, and then on that time only it will be cleared. Then there will be polls and you will need to answer these polls, and it will be very much interactive kind of session. And for all the upcoming exams, the faculty they keep on conducting the short courses and crash courses so that you are always updated about the upcoming exams and you are not feeling anything left out sitting at home. So you are always into that tempo of yes, my studies are going on. And live tests and quizzes are there, and you can retake these tests and quizzes as many number of times as you want to. So that is also one of the great benefit, because whenever you are preparing for any competitive examination, what one needs is to evaluate themselves that how much they have learned so far. So retaking quiz is that benefit that you can just go and see that how much you have learned so far. Then you will get unlimited access to the subscription. You will have so many recorded videos by so many different faculties at the same concept. If you are not able to get it from one, you can just switch and you can watch some other videos of other faculties and you will be understanding the thing very well. So that is the another benefit with Unacademy. Then there are free live classes, and these free live classes are conducted on YouTube, and they are taken as the special sessions on the Unacademy Learning app. So you can just download this Unacademy Learning app from the Play Store, and you can log in and make your account, and you will get access to all the special classes. And YouTube classes are definitely just like that available on YouTube, which are also free and which are also conducted live every time. And then there are this plus courses. So this plus course is paid, and only those who have the subscription will be eligible to watch them. They are not free, but this plus subscription is also coming in a very pocket friendly amount. What I would suggest is go for a yearly or 6 monthly plan so that you can make the most out of all the available data, 1 monthly and 3 monthly. They are also very much beneficial, but if you're going for a larger pack, it will be much better for you. And in this subscription charge, if you want a 10% discount, use my referral code FB01. So by using my referral code FB01, you will be getting a 10% discount. That is a great benefit with Unacademy and using my referral code. So today we are going to talk about the topic of this class is origin of cells. This origin of cells and unicellular organism. Unicellular organism. So this topic we are doing in two parts, part 1 and part 2. So the class is of a limited time period, that is around 1 hour. So in 1 hour, how much part we'll be able to do, that will be included in part 1 and rest we'll be doing in part 2. So talking about the origin of cells and unicellular organisms, we are going to talk it in terms of the theories given by various scientists, that the theory of abiogenesis and biogenesis, this all is going to come under the origin of cells and unicellular organisms. So first of all, Before origin of cells, let us talk about origin of life. Origin of life. So, theory of spontaneous generation was very much into being at a certain point of time on in this world. So, theory of spontaneous generation. Theory of spontaneous generation. So what does this theory says is, the another word for this theory is abiogenesis. Abiogenesis. So abiogenesis is generation of life from non-living matter. So whenever non-living matter is included, you will see the word abio, abiotic, abiogenesis. So whenever non-living matter is included, it is abio. So when non-living matter is used for the production of the alive form of life, is called as abiogenesis. So generation of life from non-living matter is theory of spontaneous generation, that no previously living organism is needed to produce the new organism. They can arise all of the sudden from the nowhere or from the physical matter. So abiogenesis is betterly these days called as spontaneous generation. So spontaneous generation is like sudden generation or without the need of precursors. Spontaneous generations and abiogenesis, this is a older theory. It is not accepted these days. Now we believe in biogenesis. So spontaneous generation, this is the oldest theory. So what does this theory says is that complex living organisms are going to get generated from the previously decaying organic substance. Decaying organic substance is the reason of production of new cells or new life. Like what profoundly they used to believe is that if there is a mice, if there is a mice in stored grain, and organisms that like mice, they can spontaneously arise in grains. If grains they are kept in a warehouse, spontaneously it will give rise to mice. Or maggots, they can spontaneously arise in the meat. What they used to believe, if we are keeping a piece of meat somewhere for a longer duration, spontaneously this meat is going to get converted into maggots, and grains they are going to give rise to mice. This was their belief, that all of the sudden from nowhere they are going to arise. This it said. Now there are certain scientists and their theories which we need to know. So first of all, let us talk about Francisco Reddy. Francisco Reddy. So what Francisco Reddy is actually, he's an Italian physician, and he was the first one who disproved the theory of spontaneous generation. So Francisco Reddy, first one to disapprove theory of spontaneous generation. So theory of spontaneous generation was disapproved for the first time. So remember, Francisco Reddy was the first person to disapprove the theory. These type of questions, the first one and name of certain scientists, these type of questions, they are frequently asked in the state level examinations. So the first one to disapprove the theory of spontaneous generation was actually told by Francisco Reddy, and he performed various experiments for disproving this theory. So what did he do? That in year 1668, Francisco Reddy, he performed experiments to check whether the maggots, they really come from the decaying meat, or they are actually present somewhere and they come and contaminate the meat and then they arise, or they feed on the meat and thus they flourish on meat. So this was his experiment. So what he did this by placing a number of jars. So in number of jars he placed n number of jars, and he covered them with the fine gauze. So some of them he covered with the fine gauze. Some of them he covered with fine gauze. So for example, 3 of them he covered with fine gauze, and rest of them he left uncovered. They were left open. So maggots, they developed only on the meat in the uncovered jar. So let us also make a piece of meat inside. So these were the jars, and the jars which were open, actually they had maggots in them after some time when they started decaying. So uncovered jars, they catered these maggots. So from this, Reddy, he concluded, so here maggots arise in the uncovered jar. So Francisco Reddy experiment. So he concluded, what did he conclude? That maggots did not come from the meat itself, but they are the tiny eggs which are laid by the flies which sit on the meat, and then the flies, they go away and these eggs, they hatch and they form the maggots. So the jars which were uncovered, they were actually attracting the flies, and those covered jars could not attract the flies and they do not have the maggots. So decaying meat do not produce maggots. Decaying meat does not produce maggot on its own. It requires eggs from flies or maggots. So this was the conclusion of Francisco Reddy. He did a very simple experiment and he disapproved the theory of spontaneous generation. So after this, the another scientist came. His name is Lazarus Spallanzani. So what was the work of Lazaro? So here let us write second. So Lazarus Spallanzani, what did he do? He was actually an Italian naturalist again. So Francisco Reddy was also Italian. He was also Italian. Reddy was a physician and Spallanzani was a naturalist. And he performed a similar set of experiment with broth. He did not use the meat, but he used broth. He used broth. There he used directly meat. Here he used broth. And he put the 2 glass flasks, sterilized them, and by boiling the flask. So he took 2 flasks and he sterilized the broth in the flask, and then what he did, one was left open and one was actually covered. One was left open and one was sealed tightly. 2 of the flasks on sterilized condition, both of them were sterilized. Then they were left like that. And then after some wait, what he observed, that the open flask was having growth and the closed one had no growth. And when it was left open, it had some growth. Growth of the microorganisms. He observed that it was starting to decay. So the other flask which was sealed out, it kept away the organisms, and The organisms, they actually came from the air. So microorganisms, they develop only in the uncovered flask. And he concluded, now the conclusion, the important thing, what did he conclude? He concluded that microorganism does not come from the broth but were in the air that entered the flask. So microorganisms enters into broth from air, and broth does not contain microorganisms. This was his conclusion. So he also ridiculed the theory of abiogenesis. Then the third one, the third scientist, so we very well know about him, that is Louis Pasteur. Louis Pasteur is a French chemist, and he finally disapproved the theory of spontaneous generation forever, and he did his experiments in the mid 1800s. Mid 1800s, he did his experiments, and he performed the same type of experiment like Lazarus, but with a little difference. He allowed the air to enter in the sterile broth as well. He did like that. So what did he do? He performed his experiment on two flasks. He took two types of flasks. One flask was with a straight neck and other with a S-shaped neck. So one of the flask was like this, normal, and other flask was with S-shaped neck. This type of neck it had, two flasks. The flask with a straight neck allowed both air and microorganisms to enter. Here it was open, so what all could go inside it? Air could also go, microorganisms could also go. But the flask with S-shaped neck allowed only air to enter. Only air could enter. But microorganisms could not enter, because if they are entering, they are going to get banged at the wall and they will stick on the wall itself and they won't be entering the broth. So broth in the straight neck flask became contaminated with microorganism, but the broth with S-shaped neck, it could not get contaminated. So contaminated, and this is not contaminated. So like what Lazarus said, that air is the one which actually takes the microorganism with it. So Louis Pasteur, he proved that air alone cannot be responsible for the production of microorganism or for decaying matter. The air should contain the causative organism also for the contamination. So air, here also air is there, but there is absence of microorganism. So not contaminated, contaminated. So this is how he said that air alone is not responsible. He concluded, what was his conclusion? That air alone is not responsible for contamination, but it requires the presence of microorganisms as well. So no microbial growth occurs in a flask with S-shaped neck, whereas microbe growth will occur in the flask which is open. And if this flask with the S-shaped neck is broken down from this place and it is left open for some time, it will have some kind of growth in it, some kind of contamination in it. So this is how he disproved the theory of abiogenesis. Disapproved theory of abiogenesis. Right. So scientists, they were finally convinced that living organism, no matter how small or how big they are, they cannot come from the non-living things, and the present theory of the living things come from biogenesis. Now the theory which is well accepted everywhere for the origin of life is biogenesis. Biogenesis. All the living organisms, they arise from a pre-existing organisms. So the theory states that living things come from the other living things. This theory says living things comes from living things. Right, this is what he said, or this is what was the conclusion from the Pasteur's experiment. So mice, they can come from mice, maggots, they can come from maggots, bacteria, they can come from bacteria, fungus, they can come from pre-existing fungus. No organism is all of the sudden appearing from nowhere. So since spontaneous generation was now proved incorrect, many scientists, they were like having this curiosity that if previously existing life form is giving rise to something, so there must be a beginning point from there where the life started. So the theory of biogenesis was actually the breakthrough point where the scientists, they started thinking that now we have to search in the direction that how the living organisms, they arise. So as soon as the theory of biogenesis came into being, scientists, they started to wonder how life is created on Earth. How life is created on Earth. So this became the curiosity. So from here, the story of the formation of Earth arises, that how the Earth formed and how many years ago they formed. So Earth, it formed around 4.5 billion years ago. Earth formed around 4.5 billion years ago. Right? So Earth formed around 4.5 billion years ago. So remember 4.5 billion, right? Billion years ago. And it is very difficult to estimate that when the origin of life actually started, but it is still believed that it began at least 3.5 billion years ago. So life started, not the actual form of today, but from the very primitive creatures. So life started around 3.5 billion years ago. Billion years ago. Right? So Earth formed around 4.5 billion years ago, and life started around 3.5 billion years ago. So the preparation of the life began a long time, that is some 4.3 billion years ago only. So life actually started 3.5 billion years ago, but there was a duration for the preparation also. So the preparative time for the life to begin was from 4.3 billion years ago only. The life it started to form. Preparative time was from 4.3 billion years. So since that time, life is fighting hard to come into being. So million of years after, under the influence of the internal terrestrial heat, solar radiation, because it was just the earth was formed newly, the atmosphere was not actually like what it is today. So atmosphere was forming, and the molecules, they were trying to have some kind of combination so that now they can reproduce on them. So all these things were actually occurring in this period. So the crucial event which actually occurred in the development of life, it occurs at the time of 3.8 billion years ago. So 3.8 billion years ago. What happened 3.8 billion years ago? That there was a formation of prebiotic soup. formation of prebiotic soup. And this prebiotic soup had all the things which is needed for the formation of new life, or all the things which is needed for development of life. So prebiotic soup formed, and this is actually theory given by A.I. Oparin. He said that the prebiotic soup is formed 3.8 billion years ago, and from this prebiotic soup, the original life it came into being at the point of 3.5 billion years ago. So the preparative for, year, or the main breakthrough point, was at the point of 3.8 billion years ago. And to solve all these doubts and queries, A.I. Oparin, he conducted experiments. So he actually did experiments on chemical evolution. Chemical evolution. He did experiments on chemical evolution. That how the chemicals they reacted and how they have formed a certain kind of life form. So the spontaneous generation, which was actually debated in the year 1924, A.I. Oparin, he said, okay, fine, I will take this pain of eradicating all the doubts of the evolution. So he proposed that life arises from the simpler molecules, and these simpler molecules are not even microscopic, they are the simple chains of biomolecules which are actually giving rise to the whole big organism. So he proposed that life arise from simpler molecules. Life arise from simpler molecules. On the lifeless earth, with a much different atmospheric condition which is actually existing today. Life arise from simpler molecules in altogether different Earth's environment. So he proposed this, that life is going to arise from the simpler, very, very simpler molecule altogether in different Earth's environment. So different in terms of what? Than today. Today it is altogether a different thing, and at that time it is altogether a different thing. However, instead of life arising suddenly, as previously spontaneous generation theory was saying, apparently he believed that it occurred over a very, very long period of time in a very gradual and slow manner. So life arose from a very gradual and slow manner. So Oparin and Haldane, there were two scientists, and they were working independently, and they proposed a theoretical model for chemical evolution. Oparin and Haldane. They both were working separately, and they gave a model for chemical evolution. Gave model for chemical evolution. This is the model which was given by them. So the Oparin and Haldane model actually suggested that there was a very reducing kind of environment. Reducing environment was there, and in this reducing environment there was production of inorganic molecules. Inorganic molecules were produced from this reducing environment, and they would spontaneously form the organic molecules. From these inorganic molecules there was generation of organic molecules, and these organic molecules are like what? They can be sugars and amino acids. sugars and amino acids. Like not the sugar which is present in today's time, but a very different kind of sugar and amino acid is produced in this reducing environment from all of the available material. First of all, they gathered and they formed inorganic molecules, and then they further changed and incorporated carbon in them, and they changed into organic molecules. So this thing actually occurred quite long before, that is 4 to 3.5 billion years ago. This thing occurred around 4 to 3.5 billion years ago. Billion years ago. This is what happened. Now, what actually happens in this case is that another scientist comes, and his name is JBS Helden, that is Helden himself. So JBS Helden, the one who gave the theory along with the apparent. So he published a paper in which he proposed. What did he propose in a paper? In a paper published by him, what did he say? He said that ultraviolet light, propose that ultraviolet light acting on the primitive reducing environment acts on primitive reducing environment with water, ammonia and methane to produce oceans that the consistency of hot dilute soup, and gave rise to hot dilute soup. This is what happens when there is a ultraviolet light, primitive reducing environment, water, ammonium, ions and methane. There will be production of hot dilute soup. The building block of the Helden's theory is the most for the reference to the hot dilute soup of the earth today. We say that earth it arised, or the life on the earth it arised on the water. So that was the hot dilute soup. Now this thing was in year 1953. Another scientist came, and his name is Stanley Miller. So along with the graduate advisory Yuri, he tested this hypothesis by constructing an apparatus that stimulated the Oparin and Helden's early Earth. So the theory they were proposed by Oparin and Helden, but it was confirmed by another two scientists, that is Stanley Miller and Yuri. So it is more commonly called as Urey-Miller experiment. So Stanley Miller and H. Yuri, they both performed the experiment related to the apparent Helden theory. So they actually justified Helden and Oparin's theory by experimentation. So they justified it by performing the experiments, and it actually gave the idea of the Earth's early hot dilute soup. So remember, Stanley and Miller, all these type of questions are, all these points are important for writing your university exam, as well as these type of questions, they come in the state level examinations, also in the SLED type of examination. Now we will be talking about the Urey-Miller experiment or Miller-Urey experiment. Miller-Urey experiment. So what happened? The Urey-Miller experiment, it is a classical experiment which fundamentally establishes the fact about the Earth's primitive atmosphere, that it was capable of producing the building blocks from the inorganic material. Inorganic material was there, and the atmosphere was reducing type, so they both combined and gave rise to the simpler building blocks of the Earth. It established that the conditions that existed in the Earth's primitive atmosphere were actually sufficient for production of the simpler molecules like amino acids and sugars. They were quite capable of producing these things. Now, what happened in the Urey-Miller experiment is they made a apparatus and they did the experiment in that apparatus. So this research was actually carried out in the University of Chicago. University of Chicago. Now, Stanley and Miller, they made an experiment and investigation on the molecular origin of life. Their innovative experimental design was such that the introduction of molecules were actually required for the formation of the whole Earth, whole life on the Earth. So they used a closed chamber, and in this closed chamber, they added methane, hydrogen, and ammonia as gases, and this environment was in a hot boiling water, and there was some lightning discharges also. And because of all these, after some days they saw that there were formation of simpler sugars and simpler amino acids such as glycine, alanine, aspartic acid, glutamic acid, and beta alanine. All these type of amino acids were formed in this type of environment, and organic acids like urea, lactic acid, glycolic acid, succinic acid, and propionic acid also formed in this environment. So Urey-Miller experiment actually demonstrated that how the biological molecules as simple as amino acid have arisen from abiotic molecules and gave rise to the continuity of the life. So first of all, we'll draw this apparatus. It was like this, a circular thing. Then there was a rod connecting it to another chamber where actually the living forms they collected, and this is how they were. It was a whole apparatus. So just ignore this overlapping line. So it was like this, and there was a detection rod over here. Then this was the tube, and here there was a thing, and this is called as Urey-Miller apparatus, and there were tungsten electrodes over here, and there were condensers over here so that the hot boiling soup can actually just condense a little bit. And then there was a valve for the release of pressure. So apparatus stimulated the synthesis of organic compounds on the prebiotic earth, and a mixture of gases were actually introduced over here. The gases were ammonia, they were methane, and hydrogen. So in this type of environment, a mixture of gases which resemble to the primitive Earth's atmosphere was fed into, and there was electrical discharge from these tungsten electrodes, and there was some kind of effect of lightning which could mimic the primitive environment, and water in the flask is reflux so that the newly formed compound is dissolved in water and they are accumulated in this flask. So the molecules which are formed, which will be, they will be accumulated over here. See over here there was a condenser, because when the gases they combine, the temperature was quite high and they could condense over here in this flask. This is condenser flask. This was there, and this flask was further evaluated that is there formation of any biomolecule or not. So compounds, they could dissolve in water and accumulate in water, and the compounds which were actually formed after some days, and this experiment they did not on a daily basis, but they did this and kept it for a longer time, and it was when it was kept for a longer time, it then generated certain biomolecules, and the amino acids which they formed, they are actually glycine, the most simpler one, then alanine, aspartic acid, glutamic acid, aspartic acid, glutamic acid. These are the amino acids which were formed, and there were some other compounds also which were formed in this experiment. So they were urea, lactic acids, glycolic acid, succinic acid, etc. So this is how they performed the experiment, that how the biomolecules they came into being. So the topic of today's session was origin of life and unicellular organism. So we have seen that how the primary requisite of the life, that is the biomolecules, how they come into being, how they are actually formed. So this experiment by Miller and Yuri successively proved that yes, life can arise from the pre-existing organism. Now, after this, the process of condensation and polymerization is important. So, condensation and polymerization. Condensation and polymerization. So the experiment of Miller and Yuri, they demonstrated the formation of simple biomolecules. But now the life, it is so complex. So how these simpler biomolecules, they gave rise to the complex biomolecules. So for understanding this concept, condensation and polymerization comes into being. So further chemical evolution, it dependent on polymerization and condensation only, that how these molecules they further joined and they form the macromolecules like polypeptides or polysaccharides. So for example, nucleic acid bases, they can be synthesized under supposed prebiotic conditions, and when they are formed, there is formation of certain nitrogenous bases, So once a nitrogen base is formed, that is required for continuation of life. So adenine is formed by condensation of HCN, a plentiful component which was present in the prebiotic environment. It was present in a very high amount. This HCN, it combined um, with the other organic molecules or sugars, and there was a catalyst for this, that is the ammonium ion or ammonia, and it gave rise to adenine. So this kind of very simple reactions, they gave rise to the nitrogenous base, which actually further led the whole process to a higher level and gave rise to polymerization. So the other bases which were synthesized were from similar reaction was actually involving some of the HCN plus water plus some of the sugars. So they gave rise to some other nitrogenous bases. This is how the nitrogenous bases they came into being. So sugars, they have been synthesized, probably no accident, then these compounds are basic component of the biological molecule. So previously just we have read in the Yuri Miller experiment that how sugars they came into being. So once sugar was there, water was there, and there was lots of HCN in that reducing environment and ammonium ion, definitely the sugar which were formed, they reacted, and that is how they gave rise to the nitrogenous bases on the fair basis of chances. So this is how the polymerization occurred. So sugars, they have been synthesized by the polymerization of formaldehyde in the reactions catalyzed by various kind of cations such as alumina or clays. So they form, and it is probably no accident that these compounds are the basic required for the biological molecules or for continuity of life, and they were apparently the most common organic substances in the prebiotic time. So they were the most common thing. You would have found lots of nitrogenous bases, you would have found lots of sugars, lots of simple acids, so water molecules, lots of ammonia, lots of HCN, lots of hydrogen. All these things were actually present in large amount in that primitive environment, and they gave rise to the complex molecules or polysaccharides, and through various kind of condensation reactions they gave rise to various types of nitrogenous bases. So this is how we have learned that how the bases they have come into being, and ammonia and amino acids we have already seen that how they came into being. Now the next thing that we need to see is proteinoids. Proteinoids. How proteinoids they came into being. So there were two scientists which were actually working together. One of was Sidney Fox, and another was from another university, and they produced synthetic peptides. They produced synthetic peptides. Synthetic peptides. So I can see some of the students who are watching this lecture live, they can write in the comment box if they are understanding the thing what I am trying to tell them, and if they are having any kind of doubt, they can ask me here only. I can see some of them, they are live. So Sydney, Fox and co-worker in the University of Miami, they did an experiment and they synthesized synthetic peptides like products of the molecular mass between 4,000 to 10,000 Daltons by heating a dry mixture of amino acids to a temperature of 150 to 180 degree Celsius. So amino acids, dry mixture of amino acid was heated at 150 degrees Celsius to 180 degrees Celsius to obtain 4,000 to 10,000 Dalton peptides. So this was obtained by Sidney Fox. So this was the point where it was quite confirming that yes, the amino acids they can join together and they can form some complex molecules such as uh, polyamino acids, or in a better term in today's time we say peptides. So these are the things, these 4,000 to 10,000 Dalton peptides which were formed, they are called as proteinoids. So they, unlike the linear proteins which are produced in today's time by the cells, proteinoids are not actually a single chain of amino acid. Like whenever we read about proteins, we know that proteins are a linear chain of amino acid, but the thing which is different between peptides and proteinoids, why we call them proteinoids actually, and what is the difference between peptides and proteinoids? So proteinoids, they are they are the branch chain of amino acid. So in a better way we should write it like branched chain of amino acids. These are proteinoids. So they were formed, and this is how we are not calling them peptides. So proteinoids, they used to exhibit different properties, different biological properties. They were having catalytic properties. So proteinoids, um, they exhibited catalytic property. Then they were digestive enzymes, and they were performing various carrier kind of activity, carrier proteins they were. So proteinoids, they came into being, and this is how the origin of organized structures they came into being. And further from the proteinoids, the further story I will be taking in the second part. That in that part we'll be talking about the co-surveys, we'll be talking about the microspheres and the first membrane cells. So these all things we'll be dealing in our next part of the same series, that is we'll be doing tomorrow. So just be there and be attentive. We'll be talking about them. And if you have any kind of doubt, you can write in the comment box. There is a live comment option also. So if you are attending the class live, you can just ask your question live, and if you are not attending live, you can ask your doubts over here. You can also write the topic which you want to study, so I'll be trying to take up that topic and explain you well. So this is it for now. We'll be meeting again tomorrow in another class of evolution. So